Okay, so the figures in this paper are rather huge, taking up half or a full or like most of the page each. So I'm going to have to break them up to better describe the process. This is figure 1a. It shows the differentiation strategy used. By modulating signal pathways that are known to regulate at least normal mouse embryonic development, the authors developed a stepwise strategy for the progression or the progressive generation of definitive endoderm, DE, hindgut endoderm, HE, and subsequently colonic organoids. So they did have access to all these types of cells from the same originating sample. And they better describe the process in the paper, but this process isn't so much important to the study as it is is it feasible or not? So they highlight it, but they don't have figures or anything about that specific process. Okay, so in figure 1b, they took bright field images of these developing human embryonic stem cells throughout that same timeline mentioned here. So, day 0, day 48, day 25, 27, you get the point. What's important to observe here is that day 38 frame. This is kind of the point at which the cells begin to take on more of that three-dimensional spherical shape that, that starts developing into like an organoid. And in frame C, we actually have that distinct colonic organoid. This is for the visual proof that the stepwise differentiation process, works. So they'll test out the spheres from day 38 and the organoids from day 48 later in the study. Okay, so now from this, the authors ran real-time qPCR analysis of colonic cell markers in, HESCs derived small intestinal spheroids, colonic spheroids, small intestinal organoids, and colonic organoids. So you have the small intestine spheres, the colon spheres, the colon organoids, the small intestine organoids. These are the day 38s, day 38s, day 48s. So don't get too confused between on the setup. The entire figure is again just measuring colonic cell markers. So this shows whether or not a given sample has traits indicative of, being like a colon cell. Specifically, instead of just some intestine that, instead of them just having a cell that's close to the target population, they want to know for a fact. And the authors throughout are basically just validating that each population is distinct, which is what they're aiming for, to demonstrate that the process does in fact make colon type cells. So eventually this figure leads into a immunohistochemistry analysis of these embryonic stem cell derived small intestine spheres, the colonic spheroids, the small intestine organoids, the colonic organoids. You have the intestine organoids, colon organoids, the small intestine spheroids. You get the point. The authors want to confirm that the expression of established colonic markers, CA2, LY6AE, CA4, in mouse embryonic colon tissue, human colon tissue, adult human colon tissue, embryonic stem cell derived. I'm just gonna say CSs and COS, but not in mouse embryonic small intestine tissue, human duodenum tissue, or ESC derived small intestine spheroids or small intestine organoids. The controls in this were mouse embryonic small intestine, and colonic, tissue, and adult human duodenum tissue. So these three are the controls. And you can see the markers clearly in every frame, which is derived from some type of colon, confirming what the authors wanted. So this is clearly a colon because you can see the red. Clearly a colon because you can see the red. Colon, so you can see colon. And the colon organoids because you can see the red. It's just confirming that again. Okay, so the authors further conducted immunocytochemistry analysis and sections of the colonic organoids for multiple markers. What they saw was that the COs adopted typical crypt-like structures in which proliferative cells at the bottom of the crypts express the marker Ki67. More than 95% of cells in crypts express CDX2, so, very bright. And around 50% express the pan-differentiation marker, keratin 20. Many of the cell types found in colonic and intestinal crypts are represented in this colon organoid, including the EPH receptor B2, and transit amplifying cells, VIA1, epithelial cells, mucin, goblin, chromogranin, endocrine cells, everything. Essentially, what the authors are confirming is that the cell types in these crypts are similar to what you would see in like an actual full atomical version, and they are. So you see the cell types, and they also went a step further, and they observed that the expected proportion of these cell types is similar to what you would actually see in like an actual colon, like epithelium. So everything kind of matches up. Okay, so figure 2, again, this is rather huge, so I'm just going to go through it piece by piece so we could better examine like every other visuals. Figure 2a. To further confirm the colonic identity of derived colonic organoids, the authors compared the transcription profiles of colonic organoids and small intestinal organoids by performing RNA sequencing. So, colonic markers previously reported in a mouse study are highly expressed in human, ESC-derived colonic organoids, while small intestinal markers are instead highly expressed in, the embryonic stem cell-derived small intestinal organoids. So this is just confirming that the small intestinal samples are not colonic samples. So these are the markers for the colon, these are the markers for the small intestine. They are different as they're supposed to be, and this is just another RNA-seq further proof that these are in fact colon-type cells. Okay. So next, the study utilized correlation data in a heat map showing sample-to-sample distances. So how far away like all these types of cells are from like where they're supposed to be, so to speak. Distance between these two samples was calculated by comparing the normalized expression data with Euclidean distances. The darker color indicates no difference, while the lighter color indicates larger differences. And unbiased clustering confirmed that colon organoids closely resembled human colon, while the embryonic stem cell derived small intestine organoids resembled like human duodenum and actual small intestine. So like they're comparing the small intestine organoids with itself over here. That's basically it. It's a correlation data, so are you seeing like how similar is that to something else? But the results kind of confirm what they were hoping for. So next, another heat map showed the expression data of marker genes of human duodenum, distal small intestine, duodenum, distal small intestine, and human colon. Marker genes of each sample type were selected such that a gene has at least 3 times higher expression in the sample type than in the other 2 sample types. So they compared human samples of these versions to like a representative embryonic stem cell, I want to say like a similar version of it from the stem cells. So duodenum would be compared to these like embryonic organoids of the small intestine. And essentially what they wanted to see is that are these stem cell derived samples similar to something that you would see in like a human sample. And while it's a bit, I guess, noisy, you do see that overall this is significantly like symmetrical, mirrored. It's a clear sample, so to speak. Okay. Furthermore, gene set enrichment analysis, GSEA, showed that the gene set highly expressed in human colon is highly enriched in the embryonic stem cell derived version. They also compared the expression profiles of embryonic stem cell derived small intestine organoids and colonic organoids with those previously reported for organoids derived from adult human duodenum, ileum, colon, and rectum. Human embryonic stem cell derived colonic organoids closely resembled the colon and rectum organoids. So very similar. And you're seeing the same thing over here. Take note that these are negative. This is like a negative scaling here. Finally, this GSEA suggested that the gene set was at least 50 times fold upregulated in adult human colon relative to the fetal colon. That is highly enriched in these derived colonic organoids.